That's the end part of this process that's happening right here for the fructose 1,6-bisphosphate. Now, what happens at this point is it actually gets split. So up until like between, I should say up to step 4, it's all been dealing with 6 carbon molecules. And then when you cut it in half, you get 2 3-carbon molecules. Okay, so this part of the slide actually takes off at the bottom, but it turns out the products from this dihydroxyacetone phosphate and then glyceraldehyde 3-phosphate, the products of these two are isomers of each other. So there's actually an enzyme called an isomerase that just turns one into the other. Okay, so what happens here is you have one molecule up here that's going through this process. It gets split into 2 different molecules, but the isomerase just gives you 2 of the same molecules. So at this point, everything's doubled. So this process, the breaking of the conversion of glucose to glucose 6-phosphate, this is a kinase, requires a phosphate, requires energy. Okay, and then a fructose 6-phosphate to fructose 1,6-bisphosphate, that requires energy as well. So another ATP gets used up here. So this is the part you need to remember. Okay, 2 phosphates get used before you break down the fructose 1,6-bisphosphate, or in the 6-carbon portion of the process, you use 2 ATPs. Okay. Then you go into the 3-carbon process, and you end up getting 2 of the same molecules, these glyceraldehyde 3-phosphates. And that's where it takes off up here. So this is the same as the end of the last slide. Okay, so at this point is when you start to get energy out of the process. And you'll notice what it says. It says all the way down there, I like this so much, it says 2, 2, 2, 2, 2. To remind you that you created 2 of the same molecule. This whole process is duplicated twice, right? Happening twice. Right, NADH gets formed. And then you're getting 2 ATPs back out. And then water. And then you get another 2 ATPs out, okay? So I'm not going to go through what all these structures are, but the net is with these 2 3-carbon molecules, the net is you get 4 ATPs out and an NADH out as well. And these are all energy molecules that can be used later, for running other processes, for example. Go ahead.
You said you get 2 phosphates, but what were the other numbers?
Oh, so in the first part? So if you have the slide, you use 2 ATPs, and then before you break it into the 2 3-carbon fragments, okay? But then you double it. So everything here is doubled because what happens is the 2 molecules you get when you split the 1,6-diphosphate, the fructose 1,6-diphosphate, when you break that apart, there's an enzyme called an isomerase, which just converts one into the other. So you end up getting this dihydroxyacetone phosphate converted into glyceraldehyde 3-phosphate. So this is doubled at this point. So now you go to here, you have 2 of these molecules. Now, if you take glyceraldehyde 3-phosphate and you convert it to 1,3-bisphosphoglycerate, all right, it gives you 1 NADH out, but there's actually 2 of these molecules going through this process. Okay, so you get 2 NADHs out, and then you get, in this process, if you just look down the process, you get 2 ATPs out here, and then 2 more ATPs out here. So you've used 2 in the first part, right? But in the later part, you produce 4, so the net is 2, yeah. And that's probably really all you have to know. No, the net gain is 2 ATPs and an NADH. Biologists are really big into this. Like, where does all this energy come from? I'm a chemist, I just eat food. Right, so, and so here, this is a nice summary slide. It tells you at which steps, okay, energy gets used, and at which steps energy gets produced. And generally speaking, when you're adding a phosphate, like you're breaking ATP up, right? That's a use of energy. So like you're adding a phosphate, glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6, bisphosphate, or if you want to think about it in your head as a diphosphate, it's a diphosphate. You're picking up phosphates. Here you have a diphosphate and you're going to a single phosphate. Right, so you're losing that phosphate bond and creating an ATP. And then you have a phosphoenolpyruvate. So this is a phosphate-containing molecule, and it loses the phosphate, and so you're getting that energy out as the phosphate gets attached to an ATP. So overall, it looks like this. Glucose, you get 2 NAD pluses going in. You end up with pyruvate, which is an important molecule. We'll talk about what happens to pyruvate. And you get 2 NADHs out, which is also, like we talked about in the last lecture, energy-containing molecules, used for the transport of electrons and hydrogen ions, right? And in that process, you gain 2 ATPs. So I had you guys learn galactose and fructose before because they're important. Turns out these molecules are also part of this process, and I will show you where they enter. But fructose, where am I at? Oh, shoots. Fructose and galactose are both converted into the liver, and fructose gets phosphorylated and gets it directly into that glycolysis process that we showed. And then galactose is phosphorylated, and then remember galactose and glucose are related to each other at 1 carbon. There's an isomerase, they call it a mutarotace, that flips that one OH over, and then it enters into this pathway as well. Okay, so if you want to see the big picture, galactose undergoes a mutarotation. So what really it's a type of isomerase. There's 4 steps, but it's really a type of isomerase that converts galactose into glucose 1-phosphate. Glucose 1-phosphate, and then it gets converted over and it becomes glucose 6-phosphate. And that's where it enters into this process. And then fructose can also enter this process by being phosphorylated, and then it gets broken apart into these 2 molecules again and enters in as glyceraldehyde, 3-phosphate. So there's different points at which these two monosaccharides can enter this glycolysis process. Oh yeah, but just like glucose, in order to get it into the process, it requires energy as it comes in. So you have to phosphorylate it before it can be used in the process. Okay, so 3 regulation points. Feedback inhibition of glucose 6-phosphate. So let me back up for a second. I don't like that one. It's too small. There we go. Step 1, hexokinase, feedback inhibition. The product here inhibits this enzyme, right? And slows the enzyme down. And also happens later on in step 3. Remember, what you're doing in step 3 is you're using ATP here, right? So if there's a lot of ATP present, it turns out it inhibits that enzyme. So high ATP inhibits the use of that enzyme. And it's also, keep touching it with my hand. High ATP also inhibits step 10. So if you go to step 10, which is down here somewhere, hang on, go up here. I have it like this. Here, this is also inhibited by high ATP concentrations, okay? So the key steps are 1, 3, and 10. 3 and 10 inhibited by ATP. 1 is inhibited by glucose 6-phosphate.
Can you go to the other page where it was
That one? That one? 8. Which one?
steps? It says
Oh, where it actually shows the steps? That one?
No, where it was right now.
Oh, sorry. Are we getting there? Oh, gotta wash my hands.
So, step one's inhibited
It's a feedback inhibition, which means the product, the glucose 6-phosphate, inhibits the enzyme. And the other two are inhibited by ATP. So if you have a lot of ATP present, you don't need to have a lot of glycolysis going on, because the whole purpose of glycolysis ultimately is to form ATP. Okay? Good? So pyruvate, what does pyruvate do? Under anaerobic conditions, under aerobic conditions, it makes acetyl-CoA. Okay? And acetyl-CoA goes into the Krebs cycle, and in the Krebs cycle ultimately produces NADH, and ultimately that produces ATP. Okay? We'll talk about the economics of it, because that's what biologists seem to really love. Sorry, it's been a long semester. I'm really tired. It's almost over. I'm a very positive person usually. I have to think, I'm just doing it for you guys. That's all I think. I was teaching another class and they canceled it. I was like, yes. And they said, but you have to teach this other class. I'm like, oh. And then they said, there's a bunch of students in it. I'm like, go. Yeah. We didn't have another teacher. Yeah. Now next semester, the crazy thing, there's like 3 or 4 of them. They all decided, hey, we should teach this class. I'm like, yeah, we've been telling you should teach this class forever. But the real problem is we don't have many instructors that have taught it before. So
it's all kind of new.
Yeah. I don't know who's teaching it. Reedley's teaching it, right?
They weren't this semester.
Yeah, they did it for years, and Miss Cornell has her degrees in biochemistry. She loves the class, but they just haven't taught it because they don't get any students. So she would be the person to take it from. I'm a chemist. I like to teach. I like to talk a lot, but that doesn't necessarily make me qualified to do everything. So, so under anaerobic conditions in humans, right, this produces lactate. And it also produces hydrogen ion. So like when you're like working out and you go into anaerobic conditions, so like you're working out hard, you burn up all your oxygen supply in your muscles because the oxygen has to be supplied by your blood. Then you undergo anaerobic oxidation, essentially, metabolism, and that produces lactic acid, okay? And the burn you feel is actually from the acidic conditions that you produce in your muscles. So that has to be cleared out. We're not going to talk too much about that. We'll mention it as we go along. Okay, so, wow, these are backwards. I just realized this whole chart should be flipped over, or I should maybe flip this over. So under there are some microorganisms, by the way. This is for the making of alcohol, right? That will take pyruvate and convert it into ethanol. That's where the ethanol comes from. I won't talk about that, although it's probably the most interesting thing on here. So under normal, right, aerobic, that means with oxygen, metabolism, what happens to pyruvate is it gets converted over to acetyl-CoA, and we talked about this the other day. That goes through the citric acid cycle, electron transport chain, and that's what produces ATP. Okay? I have the numbers on the next slide, but that produces actually, for every glucose molecule produces 2 pyruvates. For every glucose molecule, you get a lot of ATP out. Okay? Without the ability to go through the citric acid cycle, the Krebs cycle, lactate produces very little energy, okay? But it's fast. It's like 100 times faster than aerobic metabolism. I'm going to skip this. All right. Yeah, so let's see. Yeah, so complete oxidation of one glucose molecule is 38 ATP molecules, okay? You only get about 2 out when you do anaerobic.
Every book has like a
What's that?
Is there like a specific number that we should follow?
38? Is that what you said? 38? Yeah, I think 38's like the number. I remembered 30 is what's on the slide. So.
I remember last semester was 32 and 36 of the others.
Yeah, and I think it kind of depends on what happens after the initial oxidation of the molecule. But I don't.
So should we follow 30 or there's?
Yeah, follow anything between 30 and 40. Yeah, that is the range. I remember 38, like when I learned it, 38 sticks in my head. But that was like 38 years ago. And as I get older, the number changes. Right? All right, so let's see. Yeah. So in some, but there's, I mean, they're pointing to that difference here, and it depends on the type of cell and exactly how that electron system works. Like we had the 4 complexes in the electron transport chain. Exactly how that works, you can, like they say here, you can get 32 or 30. And like I said, I remember 38 and some other numbers. So I think 30 to 40 is a good ballpark range to remember. Okay. Okay, so we're going to talk a little bit about glycogen, because glycogen is how sugars are, glucose monomers are stored in our body, okay, as she drinks her sugary Dutch Brothers drink. That's a huge drink. Is it good? Is that good?
I don't know, you guys ever try the 911?
The 911? Is that like a lot of caffeine?
It's like 6
Oh my god.
Oh my gosh. I did that once. Once. I went to Scotland and I stayed in a place that had, maybe I was in Manchester for that one. I stayed in this hotel chain because, anyways, they had free espresso in the morning, like a machine. You could just walk up to it, hit double, 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 double. And I thought, oh, this is fun. I went on the fastest, longest walk I'd ever gone on after that. My roommate, the guy I was traveling with, got really sick. So I'm like, well, what am I going to do? I'm not going to go out and goof around. He's too sick. So I went out and walked a few miles really fast. Yeah. So anyways, so in our muscles, we store glycogen. We also store it in some other tissues. But glycogen is, when we talked about amylose and amylopectin, right? What's amylose? It's that long, single-chain polymer of glucose, right? It's linear. Amylopectin has branches. It's linear. It's branched 1 to 6, right? The only difference between that and glycogen is the branching occurs more frequently. So where they'll say things like, oh yeah, like it depends on what you read again, books.